Hello, dear all. Hope you feel good. My name is Fernand. I'm a research assistant at the Laboratory of Genetics, Horticulture and Seed Science, Faculty of Agronomic Science from the University of Abomey-Calavi, country of Benin. Here, I would like to share with you a review research on the molecular mechanisms and of seed dormancy in plants, an overview and implication for traditional leafy vegetables. So, as you know, there are more than 1,000 species used by African rural communities for dietary diversity, for medicine purpose, for food tradition, and cultural identity. So, the key bottlenecks limiting an all-year-round production were the loss of seeds viability, poor and delayed germination, and inaccessibility of farmers to high quality seeds due to the factors such as harvest time, storage, and conservation conditions of seed, as well as seed dormancy. While other factors can be easily controlled, breaking seed dormancy requires a full knowledge of the seed intrinsic nature and physiology. So we engage this research to synthesize the scattered knowledge on seed dormancy constraints in traditional leafy vegetables, to highlight the main seed dormancy regulation factors, and to develop a conceptual approach for molecular genetics analysis of seed dormancy in traditional leafy vegetables. In the first time, we reported the occurrence of various kinds of seed dormancy, basically primary dormancy, physical and physiological dormancy among traditional leafy vegetable species, from mature, freshly harvested seed to the dry seed for species like Talinum triangular, Solanum nigrum for Cocorus olitoris, and for Gynandropsis gynandra. In the second time, we found that seed dormancy is regulated by phenotypic and environmental factors during the seed development, during the after ripening, and during the seed germination. So the different non-regulator factors involved in seed dormancy control and direct action are presented in the figure 1. So we can find that abscisic acid and gibberellic acid are basically the main hormone that decide if seed will germinate or not. So the environment factors such as after ripening, stratification, light, and seed components influence also seed germinability. However, the bulk of knowledge was based on cereal and Arabidopsis, and there is little awareness about seed dormancy facts and mechanisms in traditional leafy vegetables. So to successfully decipher seed dormancy in those species, we used Gynandra to illustrate possible research avenues and highlight the potential of these species as a model plant for seed dormancy analysis. So spider plant is a promising vegetable species for food security and income generation for African communities. Its genome size is relatively small, with the possibility to obtain a fruit generation per year, offering many options in terms of breeding strategies. Also, Cleomaceae family and Brassicaceae family are closely related with faster molecular evolution rates, and seed dormancy is strongly reported in spider plant, and spider plant's accession should differ largely in the seed dormancy behavior. Then the proposed research avenue is presented in figure 2 here, and knowing that the World Vegetable Center, the Laboratory of Genetic, Horticulture and Seed Science, have a strong correlation for Gynandra. A further research is needed to the identification of major genetics and the molecular factors underlying seed dormancy during seed development, seed storage, and germination, so that we can develop the new cultivars that farmer can effectively use. So multidisciplinary research teams, including physiologists, geneticists, and bioinformaticians, are required to quickly and efficiently make significant progress toward breeding for non-dormancy in traditional leafy vegetables. So that is my presentation. I'm at the end. So thank you.